All right, so our first topic for week 7 is muscle anatomy. So the learning objectives for this lecture are understand the similarities and differences in the 3 different types of muscle, which are skeletal, cardiac, and smooth, in terms of their structure, function, distribution, and innervation, and then be able to explain the concepts of the sarcomere and the sliding filament theory. So starting off, this is going back to your basics of anatomy. So there's 4 different primary tissue types. We've got epithelium, muscle tissue, nervous tissue, and connective tissue. So I'm not sure which ones you've already looked at. Muscle tissue might be the first one you're starting off with, but essentially there are 3 different types of muscle: skeletal, cardiac, and smooth muscle. All right, so the function of muscle tissue, this is for all 3 types. We've got produce movement of the body, limbs, organs, tubes, and glands, maintain posture and body position, maintain body temperature through thermoregulation. For example, you've got shivering and vasoconstriction, support soft tissues in the organs, guard and restrict entrances and exits to organs, for example, sphincter control, and store nutrient reserves in metabolic tissue. So the key properties of muscle tissue, to remember this, it's C. So we've got contractility, which is the ability to shorten actively and exert pull or tension that can be harnessed by connective tissue. Excitability, which is the ability to respond to stimulation. Extensibility, which is the ability to continue to contract over a range of resting lengths. And elasticity, the ability to rebound towards its original length after contraction. So like I've said previously, we've got the 3 different types of muscle, which we're now going to go into depth into. So the first one is skeletal muscle. So referring to the diagram on the right, we've got 3 different layers of connective tissue in skeletal muscle. So around the outside, we have epimysium, which covers the entire muscle and separates it from surrounding tissues and organs. And the next level down, we've got perimysium, and this divides the muscles into a series of compartments, which we call fascicles. Each fascicle contains blood vessels and nerves. Then the next layer down, we've got endomysium, and this surrounds individual muscle cells or muscle fibers. Each muscle fiber is like its own individual little unit. And we say that skeletal muscle is under somatic voluntary control. So that's referring to the innervation. I'm not sure how much you guys have done on the nervous system yet, but essentially skeletal muscle is the only one out of these 3 types that we have voluntary control over. So we've got some special skeletal muscle terms. So it's like a different language essentially. So first, when you say fiber, that means an individual muscle cell. Myofibrils are found inside each muscle cell or fiber. And the myofibrils contain 2 different microfilaments, actin and myosin. We'll discuss those later. Sarcoplasm is cytoplasm, but for a muscle cell. Sarcolemma is the cell membrane for a muscle cell. Sarcoplasmic reticulum is the equivalent of smooth endoplasmic reticulum, which is found in muscle fibers. And sarcomere is the smallest contractile subunit, which we're going to discuss next. So on the right here, we've got a histological image of a skeletal muscle cell. So these individual muscle cells can be up to 20 centimeters long, which is quite large compared to other cells in the body. And they can contain up to 1,000 nuclei per cell, which is also a lot considering most normal cells have only 1 nucleus in each cell. The striations, which you can see, they're like the pink and dark pink stripes in this image. Those are present due to the presence of myosin and actin microfilaments. So we have some darker lines and some lighter bands, and we'll discuss why that happens next. But essentially, if you see striations, that means it's characteristic of a skeletal muscle cell. And skeletal muscle cells also contain well-developed sarcoplasmic reticulum. So that's essentially smooth endoplasmic reticulum and T-tubules. So a sarcomere. So we've got the 2 diagrams on the right here. So the first one is of like an in-depth image of a sarcomere. And then the second one just breaks it down into all of the different lines and bands that you're going to hear about. So a sarcomere is like a unit of measurement, and it's the distance between 2 Z lines. And now we've got two different bands inside the sarcomere, the A band and the I band. The A band has both thick myosin filaments and thin actin filaments. So actin is always thin and myosin is always thick, whereas the I band only has thin actin filaments. So if you think back to the image from before where we had the dark purple lines and the light pink lines, the light pink is the I band and the darker one is the A band. Then the T tubules, they are present at the AI junctions. So where you have the A and the I bands intersecting, you'll have a T tubule that comes down. And T tubules are just invaginations of the plasma membrane. So you've got the plasma membrane, which is straight like this, and then this is a T tubule where it folds in on itself. So like I was saying, T tubules, they allow for electrical impulses to pass deep into the cell to increase the speed of muscle contraction. So it's kind of just increasing the surface area of the cell membrane in a way, because you've got it over the surface of a muscle cell, but now it also goes deep into the muscle cell. So that just allows everything the speed of contraction to happen at the same time throughout the cell. T tubules are surrounded by sarcoplasmic reticulum on each side. And those parts of the sarcoplasmic reticulum are called the terminal cisternae. And if we look at the whole combination of the T tubule in the middle and then the sarcoplasmic reticulum on each side, we call that structure a triad because you've got three different parts. So the stages of contraction of skeletal muscle. The first step is an action potential, which we discussed last week, arrives at the neuromuscular junction, which is also called the motor end plate. That's where you have a neuron that meets a muscle, so neuromuscular junction. And this causes local depolarization of the sarcolemma. So that's the plasma membrane of the cell. Then the depolarization travels down through the T tubule system. Calcium is released from the sarcoplasmic reticulum into the sarcoplasm of the cell. So the sarcoplasmic reticulum, if you remember, is like the endoplasmic reticulum, and that's where calcium is stored. And the sarcoplasm is like the cytoplasm of the muscle cell. So essentially calcium just diffuses throughout the cell. Calcium diffuses to the myofilaments. The myofilaments are your actin and myosin, where it binds to the troponin C portion of the troponin complex. So the troponin C is a part of the actin and myosin, but you don't need to worry about it too much because you go a lot more in depth into this next semester. So actomyosin cross bridges, that's between the actin and the myosin, they are formed. Repeated cycles of this cross bridging between myosin and actin filaments shortens the length of the sarcomeres. And essentially that is how a contraction happens because the length of the sarcomere changes. So if you can imagine, the muscle contracts. When the action potential ends, calcium returns back to the sarcoplasmic reticulum where it's stored, and this terminates the actin-myosin interactions, and the contraction ends. So next we've got cardiac muscle. So cardiac muscles are branched cells with one central nucleus. They can sometimes have two, but they certainly don't have 1,000. They also contain striations and sarcomeres, just like skeletal muscle. Cardiac muscle cells are joined end to end by these things which we call intercalated discs, and this helps the muscle cells communicate with each other. Intercalated discs contain gap junctions, adherens junctions, and desmosomes. So those are just three different types of, essentially they enable communication. They're just three different types of structures that all enable cells to communicate in different ways. Cardiac muscle cells are abundant in mitochondria due to their high levels of aerobic metabolism, because if you can imagine, cardiac muscle cells are obviously found in the heart because they're cardiac muscle, and the heart needs a lot of energy for all of the contracting that it does to pump blood around the body. Cardiac muscles are under pacemaker involuntary control. So as opposed to skeletal muscle, which is controlled by the the somatic nervous system, which means we can contract our skeletal muscles whenever we want. The cardiac muscle, you don't have to think about it. It's not something that you say you're going to do and then it happens. It just happens involuntarily. And finally, we've got smooth muscle. So smooth muscle consists of spindle-shaped cells with a central nucleus. They will only have one nucleus. Cells are usually joined to each other by gap junctions or electrical connections. And smooth muscle doesn't have striations or sarcomeres. They contain well-developed sarcoplasmic reticulum, but they don't have T-tubules. Smooth muscle is usually found lining hollow tubes or organs, including the esophagus, the bladder, the vas deferens, the bronchus, the aorta, and the uterus, and in the smaller arteries and sweat glands. And smooth muscle is under autonomic involuntary control. So again, you can't contract your own smooth muscle. That's something that happens without you thinking about it. So next we have the muscles and blood vessels prac. I'm not sure if this is in the same format for you guys as it was for us last year, but it's like an online workshop that you just go through in your own time. All of the content that you need to know for the muscles part of it is what we've just covered in that lecture. So we're just going to discuss the blood vessels part of it now. So essentially for blood vessels, I've got a little summary here of how your blood moves around the body. So if we start out in the heart with deoxygenated blood, first that's going to travel through the pulmonary arteries. Arteries always travel away from the heart. So pulmonary arteries are leaving the heart and going towards the lungs. So they carry your deoxygenated blood to the lungs where the blood is oxygenated. So that's why it's turned red now in my little diagram. And then the oxygenated blood leaves the lungs through the pulmonary veins. Veins always carry blood towards the heart. So then once the oxygenated blood is back in the heart, then it leaves in arteries to go around the body. Capillaries is where the blood diffuses out and becomes deoxygenated again. So that's where the body takes up that oxygen. So then it's back to blue, it's deoxygenated, and it returns back towards the heart in veins, where this process can repeat. So that's the cycle of blood in the body. Functions of blood vessels, there are numerous, but some of the main ones are listed here. We've got gas exchange, so obviously allowing the body cells to take up the oxygen, temperature regulation, pressure regulation, fluid regulation, and immune cell activation. Layers of a blood vessel. So as you can see in the diagram to the right, we're going from innermost to outermost now. So this is the inside of the blood vessel. We have the tunica intima, which is made of endothelial cells. Endothelial cells are just like your epithelial cells, but the prefix endo means like to do with blood vessels. So endothelial cells are only found in blood vessels. Next, we have the subendothelial layer, which is one out from the tunica intima. Then we have the internal elastic lamina. Next, we have the tunica media, which is a muscular layer. Then we have the external elastic lamina. And finally, we have the tunica adventitia, which is an elastic layer. So arteries, the purpose of arteries is to carry blood away from the heart. Arterial walls contain more elastin than veins. And they're much thicker. So when you start doing dissections next semester, you'll be able to see arteries and veins within the body. And one way to recognize an artery is that it will always have a much thicker wall than the veins. And the lumen size, the lumen is just the gap in the middle of a blood vessel, like the hollow part. Arteries do not contain valves. And as a general rule, arteries carry oxygenated blood, except for the pulmonary arteries, which are taking deoxygenated blood from the heart to the lungs, and the umbilical arteries, which take deoxygenated blood from the fetal circulation to the placenta. Types of arteries. There are 3 different types of arteries. We've got elastic arteries, muscular arteries, and arterioles. And as you can see in the diagram to the right, they're going away from the heart in that order. So elastic arteries are found closest to the heart, and arterioles are found the furthest away from the heart. So elastic arteries are the largest and closest to the heart, and they contain lots of elastic tissue in the tunica media, so that's that middle layer, and this helps to combat pressure stress from the heart. The muscular arteries are mainly composed of smooth muscle in the tunica media instead of elastic tissue, and they are responsible for increasing blood distribution. Finally, the arterioles are the transition between arteries and capillaries. and their walls are only composed of a few layers of smooth muscle cells, and they're the primary site of vascular resistance. The veins carry blood towards the heart. They have much thinner walls and contain less elastin than the arteries. Many veins contain valves, which stops blood from flowing back in the opposite direction and helps to essentially pump it back towards the heart. Veins generally carry deoxygenated blood, except for the pulmonary veins, which are carrying oxygenated blood back to the heart from the lungs, and umbilical veins, which are carrying oxygenated blood back to the fetal circulation from the placenta. And finally, capillaries. So this is the site of oxygen and nutrient exchange. It's where the blood loses its oxygen, essentially. So their walls contain only endothelium and basement membrane. Red blood cells need to be able to squeeze in and out. We've also got all of the white blood cells that can move out of capillaries and a lot of fluid. So basically, they need to be really thin walls to increase their permeability. And the lymphatic system. So this isn't quite blood vessels, it's not on the same topic anymore, but it's also found as part of that prac. So lymph is fluid that contains white blood cells, proteins, salt, glucose, fat, and water. It's found everywhere in the body except for the teeth, bone marrow, and the central nervous system. The functions of the lymphatic system include drainage and filtering of extracellular tissue fluid, recognition of foreign antigens, priming of immune cells for defense. The lymph nodes, so these are little nodules that are made up of lymphatic tissue, and they're basically stops on the way for the lymph moving around the body. So every time lymph passes through one of these lymph nodes, the lymph is filtered out. So any antigens can be taken out of it and taken up by the immune cells. In the lymph nodes, cell debris is removed, foreign particles are filtered, and foreign proteins are recognized. This is kind of just an intro to the lymphatic system because you'll discuss it a lot more when you start doing immunology. And that's all from me.
Hey guys, I'll just be taking you through cancer today. So looking at the biochemistry of cancer, I'm thinking about characteristics and behavior of cancer cells, describing differences between benign and malignant cancers, as well as the effects of localized and metastatic cancer. We'll look at different types of mutations, as well as, in a more clinical sense, the effects of cancer on a patient. So yeah, really important stuff we'll be covering. Just beginning with characteristics and behavior of cancer cells. So they're resistant to inhibitory growth signals and apoptosis, and they can also produce their own growth factors. And so this means that cancer cells do not exit the cell cycle, whereas other cells do. And you can see that if they don't exit the cell cycle, this can lead to kind of uncontrolled replication of these cancer cells. They lack contact inhibition. Often you'll have an accumulation of genetic errors and chromosomal abnormalities. They've got reduced requirement for growth factors. We call them immortal sometimes because cancer cells can be self-sustaining, and often they're self-sustaining through a process known as angiogenesis, which is where cancer cells can grow blood vessels from existing vasculature. They're able to escape immune surveillance by mimicking other body cells. Something that's super important is that they have active telomerase. So active telomerase is an enzyme which replenishes the telomeres, and telomeres cap chromosomes and they indicate the age of a cell. And when the cell divides, these telomeres shorten. So cells whose chromosomes lack or have very short telomeres will undergo apoptosis. But because cancer cells have active telomerase, they can replenish their telomeres and therefore avoid apoptosis. And because of that, again, they won't exit the cell cycle. So important enzyme here. Often you'll find that cancer cells have an enlarged nucleus and little cytoplasm. And this is kind of predicted because you've got all this repeated mitosis of cells, this uncontrolled cell replication. This leads to accumulation of DNA, which can give off the image of an enlarged nucleus when it is stained under a microscope. And then just lastly, you've got loss of specialized features, often because cancer cells are immature cells, which I go into just a little bit more detail soon. So here we've got an image of normal cells on the left, and then more cancer-like cells on the right. So looking at this first, this second column, first row here, we can see compared to a normal cell, they've got a much larger nucleus because of the accumulation of DNA. And we've got nuclei that aren't necessarily the same shape. They can vary. Just beneath it, we've got lots of cells here that are disorganized compared to the more organized arrangement on the left-hand side. Again, variations in size and shape, and then loss of normal features. So we can clearly see kind of some of these specialized features in the normal cell, but we lose that distinction when these cancer cells are stained. So benign versus malignant cancers. So benign cancers do not spread because there's a fibrous capsule surrounding the cells. Because of this fibrous capsule, the benign cancers remain localized at the site of origin. So this is one of the key distinguishing factors, being localized at the site of origin. Often they'll resemble the normal tissue they came from, and usually benign cancers don't have a high mortality rate. They're not fatal unless they're growing in confined, vulnerable spaces, particularly the brain. But usually benign cancers have a good outcome. In comparison, malignant cancers have a high rate of metastasis and invasion of surrounding tissue. So metastasis refers to the spread of a tumor to a site discontinuous with the primary tumor. So this can be local or distant, and is a key indicator of a malignant cancer. So yeah, they can invade distant sites. Often they won't resemble the tissue or cell of origin, and we call this anaplasia. So this is just kind of a lack of differentiation. They don't look like the tissue that they came from, and can often be associated with quite fatal outcomes. So yeah, malignant cancers are dangerous. Looking at varying levels of benign cancers compared to malignant, you can see that cancer cells to begin with stain much darker compared to normal cells. This is because you've got this excess of genetic material. And you'll notice that as a cancer moves from being benign, from normal to benign to malignant, we've got this abnormal and inconsistent size and shape of these cells in this kind of disordered arrangement with huge nuclei. We call this pleomorphism. So if you see the term pleomorphism, it's just referring to the abnormal and inconsistent size and shape of the cells. But yeah, we can just see this huge nucleus here in the disordered fashion, particularly in the malignant one as well. So looking at localized versus metastatic cancer. So localized cancer will often involve a specific type of cell, and these cancer cells replace the normal cells. These cancer cells can often be immature, and this is a result of they haven't had sufficient time to develop during the cell cycle. They just keep replicating, they keep dividing. Because of this, if cancer cells are immature, they can lose their specialized features, and therefore are unable to complete their specialized functions, and so are therefore dysfunctional. We've got competition for nutrients and blood supply. This can often result in things like rapid weight loss, looking at a more clinical symptom, and then they can displace healthy neighbor cells. Metastatic cancer, in comparison, will spread via blood vessels and lymphatics. They don't need to have any anchorage anywhere, and often they can be dormant for long periods of time. And they can be dormant because of lack of angiogenesis. So angiogenesis can be a really long process to develop because you're growing new capillaries. The longer this takes, the more dormant the cancer will be. The stages of metastatic cancer. So you've got cancer cells secrete enzymes which break through the capillaries. Because of this, the cells can now enter the bloodstream and the lymph, and they will flow along blood and lymph patterns and then adhere to walls of capillaries and exit into tissue. At this stage, I don't think you guys need to know in too much detail where different cancers spread to, but it's important to know that the lymphatic system can play kind of a cardinal role in the spread of cancer. Okay, thinking about the development of cancer. So damage to DNA that can be inherited is often 10%, or it can be environmentally acquired. So this might be through things such as smoking, radiation, viruses, or cancer can develop as a result of the accumulation of genetic errors that promote the growth and longevity of cancer cells whilst compromising repair mechanisms. Usually in terms of genetic errors or mutation, we require at least 4 to 6 in a cell. So it's not just, it would be incorrect to think that one mutation can cause cancer. It's a result of multiple mutations. And these mutations can occur in oncogenes, tumor suppressor genes, or mismatch repair genes. So these three. And we'll go into a bit of detail about each of them now. So oncogenes. So with proto-oncogenes, they code for proteins that support growth, division, and cell adhesion. So these are good. A mutated proto-oncogene is then called an oncogene. So this is a gain of function mutation, which is dominant. And really important here that you remember this, it's a one-hit hypothesis. So a mutation in one allele is enough to cause cancer, and it may promote or predispose you to cancer development, and it stimulates premature mitosis and acceleration of cell growth. So key takeaway, it's gain of function and one-hit hypothesis. This is a fairly high yield question you'll get. Looking at tumor suppressor genes, just straight away noticing rather than a one-hit hypothesis, they are a two-hit hypothesis mutation. So it requires at least 2 mutated alleles to promote cancer. And rather than gain of function with oncogene, you've got loss of function. So tumor suppressor genes usually suppress the activation of oncogenes, which can cause cancer, or they suppress the activation of preventing cell division. So specifically, they control cell cycle checkpoints. And in some examples of tumor suppressor genes are retinoblastoma, so RB, and P53, and these work at the G1/S checkpoint. So they've got no time to repair DNA and it allows for evasion of apoptosis, unregulated cell growth. So this is what happens when you get mutations in tumor suppressor genes, and these are bad. With mismatch repair genes repair mutated DNA during the S phase. Again, it's a two-hit hypothesis and requires at least 2 mutated alleles to cause cancer, and it's a loss of function mutation. On the more clinical side, so effects of cancer on the patient. So it displaces normal adjacent tissue and can block vital passages such as vessels or organs. Often tissue function would deteriorate. And again, linking back to the characteristics of cancer cells, this is because cancer cells are immature and cannot carry out their specialized functions, and so cannot carry out the function of the tissue. So that's why that deteriorates. You've got high metabolic requirements, which stimulate the breakdown of other cells. So proliferation of cancer in general is extremely metabolically demanding because it competes with other normal cells for nutrition and blood supply. And as a result, you can get excessive weight loss and breakdown of other cells. And then often you might find a swelling mass, which may be indicative of a blockage in the lymphatic system. And in this example here in the image in the bottom right, we've got swollen cervical lymph nodes. Okay, taking a dive into inherited cancer characteristics. So several close or first degree relatives with a common cancer or related cancers may predispose you to an inherited cancer of some form. 2 members of the same family with the same rare cancer is something to look out for. Other things such as early age of onset and bilateral cancer in paired organs. So paired organs refer to organs that come in two. So for example, kidneys, not like the heart, for example. So bilateral cancer would mean cancer in both of your kidneys, left and right. This is something to look out for. And then tumors that are in two different organs within the same individual. And this is unlikely to both randomly mutate. So it's important to keep an eye out for this as well when thinking about inherited cancer characteristics. So yeah, that brings us an end to cancer for this week.
All right, and I'm just going to cover Pharm. So this is quite a short lecture for this week. It's just really an introduction to what the study of pharmacology is. So that being said, pharmacology is how pretty much a drug interacts with the living system. So there's two terms that you'll hear a lot. That's pharmacokinetics and pharmacodynamics. So pharmacokinetics is what the body does to the drug, and pharmacodynamics is what the drug does to the body. So how drugs bind to their target sites, how this binding will then go to a biological effect. So you'll hear these phrases mentioned quite a bit in your upcoming lectures. Next up. So this is pretty much just the phases of drug action. So there's three main phases. So the pharmaceutical phase is when the drug is taken from the package. The pharmacokinetic phase is when the body is doing its stuff to the drug. So the body absorbs and distributes, metabolizes and excretes the drugs. So that this pharmacokinetic phase I think will be covered next week for you, and then the pharmacodynamic phase will be covered for the rest of the semester. So that being said, pharmacodynamic phase is where the drug pretty much has its effect on the body. So it binds to its target site and then it leads to its effect. Next up, what is a drug? So a drug is pretty much just a compound which is administered to produce an effect to the body. So that could be physiological or psychological. And it's also biologically active in small quantities. So you don't really have a massive pill to take when you take a drug. It's quite a small quantity, so quite powerful. This can include, you have your drugs of abuse, you have your therapeutic substances, toxic substances, and endogenous substances. And drugs can be either natural, either plant-derived or natural, or synthetics. That's synthesized from chemicals. Next up, so we've got a few different ways to name drugs. So the generic name of drugs is pretty much just the active chemical ingredient. So for example, you've got paracetamol, and that's your active chemical ingredient in Panadol. So instead of being called Panadol, it's just called paracetamol. So this is just your brandless drug. Then you've got your chemical name, which is how you name the drug based on its structure of chemical compound. You won't really learn the chemical names very much. You'll mainly learn the generic names and the trade names. So the chemical name is just really very much the scientific name which the pharmacologist will use. And next you have your trade name. So your trade name is your name given by a pharmacy company. So for example, you've got your Panadol, you've got your Nurofen, and Panadol is really just paracetamol but with its trade name. So next up, we've got receptors. So receptors are what a drug binds to. Your receptor can either be intracellular or extracellular, and it's responsible for transducting a signal and also communicating cell to cell. So it interacts with the drugs and initiates a physiological response. And receptors are important for specificity because it means that there's action at the specific site where the receptor exists. So where the drug's target receptor is, you'll have action. Where it isn't, you won't have action. So that's why receptors are important. But receptors are also important for selectivity, because say, for example, you've got receptors that are scattered throughout the body, but they're the same receptor, and the drug has an affinity for that receptor, then that means that the drug will act wherever those receptors are around the body, if that makes sense. So it's important for it to select where to act. Next up. So just diving quickly into specificity and selectivity. Specificity is that a drug will act on a receptor only found on a specific organ. So for example, a drug might only target the beta receptors on the heart, and that's it. It won't target the beta receptors anywhere else, just on the heart. And selectivity is that what I was explaining before. So wherever that receptor is, that's where the drug will act. For example, one could be beta receptors this time, but not just on the heart, but beta receptors wherever it's found in the body. So, and these are important terms to know. The specificity is only on a specific organ, and selectivity is just wherever the receptor is. Next up, we have a quick overview of targets of drugs. You really don't need to know much about this. As I've said before, the Intro to Pharm lecture is very short. It just wants you to know really your basics about drugs and some basic terminology. So targets of drugs. Drugs usually target proteins, receptors, whether they be adrenoceptors, nicotinic or muscarinic receptors. It also targets ion channels, it targets enzymes, and it targets sometimes carriers or transporters. Next up. So receptor-drug relationships are important though for three main reasons. So they help us figure out the concentration of the drug required for the effect, because once we figure out the affinity of a receptor for a drug, then we can figure out how much of that drug we need to be able to get an effect. For example, if there's a really, really high affinity for a receptor for a particular drug, we'll need a lower dose. But if there's a low affinity, then we will need a higher dose of that drug. Additionally, the selectivity of drugs is also determined by receptors. So receptors can have various affinities for different classes. Drugs can have various affinities for different classes of receptors. So this influences the therapeutic and toxic effects, because sometimes drugs can have affinities for multiple receptors at once. So it's important to keep that in mind as well. So you want to try and, if you're trying to just target one particular organ, you want to choose a drug that has a very high selectivity and specificity. So it'll only target that organ and that receptor, if that makes sense. You don't want to do one with a broad selectivity necessarily, because then you'll be targeting, you'll be targeting receptors all over the body you might only want to target one particular organ. And additionally, the receptor determines, And additionally, the receptor determines if a drug is an agonist or an antagonist. So if the drug binds to the receptor and then activates the receptor, it's an agonist. But if it binds to the receptor but cannot activate it, it's an antagonist. So this is also known as the lock and key model. So lock and key is agonist. So the drug binds to the receptor, it can open the key, it can open the lock. The key can open the lock. So therefore it's an agonist. But in an antagonist, it binds to the receptor, the key can't open the lock, and it's an antagonist. So it stops the lock from being opened. Next up, we have transmembrane signaling. So this you really don't need to know very much at all. I don't think it's covered in the lecture, but it's a little bit more information. There are 5 different kind of methods whereby transmembrane signaling can be achieved. So you've got your hydrophobic ligand, which crosses the plasma cell membrane and then acts on an intracellular receptor. So that's in the image number 1, what you can see. Next up, you've got a receptor whose intracellular enzymatic activity is regulated by ligand binding, and it changes its conformation. So if you can see in image 2, you've got the ligand, which has which is bound to the receptor on the outside, and then if you see the intracellular activity is regulated, so you can see A going to B once the ligand has bound. C is a transmembrane receptor, which stimulates a protein kinase when activated by the ligand again. 4 is a ligand-gated receptor. So if you see that, this little blue thing, oh, by the way, the little blue circle is what we call a ligand. So this little blue circle has just approached the receptor. If this approaches to the receptor, it will open the receptor. If it goes away, it will close the receptor. And then finally, we have a receptor protein. So the receptor protein stimulates a G protein and then activates a lot of different intracellular messages. So if you see the ligand's bound to the receptor there, and the G protein has then bound to another protein, and then so it's kind of the G protein is a messenger from the protein to the next protein, if that makes sense. Next up, one of your learning objectives is just to know some sources for key drug info. So I'm just popping this up here. Feel free to return to it if you need it. But just so you know, these are what we view as reputable resources for key drug info. And you will come back to this a lot, especially when you're doing micro later on in the semester and next semester. So I think that is it for me.
Okay, so this week we'll be going through blood components and haematopoisis, which is pretty much how blood cell components are made. So first thing, just overview of blood cell components. Majority of your blood is plasma. So that's going to contain things like water, proteins, metabolites, and nutrients, pretty much anything that your blood needs to be circulating around your body except for oxygen. because oxygen will be transported by your red blood cells, which we put there as 45% of your blood is red blood cells. I'll go into that in a bit more detail later. Sorry, Jay, can you go back? And then just a really small proportion of your blood is something called a buffy coat. And that's what contains all your immune cells and your platelets. Okay. So hematopoiesis or hematopoiesis is pretty much the formation of blood cell components. And we'll go into all the different blood cell components and how they're made. But in general, the key requirements there are iron, vitamin B12, and folic acid. So all cells start with as a hematopoietic stem cell, as you can see at the top of that diagram, and then they'll split into a common myeloid progenitor and a common lymphoid progenitor. Your common myeloid progenitors, they become your platelets, your red blood cells, and they become some of your white blood cells. The white blood cells they become are mainly ones of the innate immune system, which you will look at specifically in a few weeks. But just know that myeloid progenitors, in terms of white blood cells, it's usually cells of the innate immune system. Whereas with your common lymphoid progenitors, that's mainly white blood cells of your adaptive immune system. I think with this diagram, it's important to know where things are happening. So the diagram shows that the initial stages happen in the bone marrow, but then you kind of have the end cells down the bottom forming in your circulation. Okay, so erythrocytes or red blood cells. Three big things about it you need to know are its appearance, its energy production, and its function. So in terms of its normal appearance, it's a biconcave disc, and that's really important for both flexibility. So it should be able to go through small spaces, small capillaries, and also to increase its surface area so that it can carry as much oxygen around the body as it can. It's also anucleate and it has no organelles. So that's also important because that just leaves more room in the cell to carry hemoglobin as opposed to a nucleus or organelles. Energy production, a key thing is that it has no mitochondria. So the ways that it produces energy are through these two pathways. I've put the names there. There's a main and a minor one, but they're not very important to know the names of. Just remember that the energy production is anaerobic in red blood cells. In terms of the function, you're probably already familiar that it's to transport gases, specifically oxygen and carbon dioxide. And don't need to know the number, but just for an idea, there's about 64 million hemoglobin molecules. So your red blood cells are carrying a lot of oxygen around your body. So erythropoiesis, that is the formation of red blood cells or erythrocytes. So we looked at the diagram on the top left a few slides before. What we're focusing for erythropoiesis are these first few steps that I've circled there. If we zoom in on those steps, you can see how the hematopoietic stem cells become red blood cells over 3 to 5 days. The important thing is to kind of know how it's regulated. So that's regulated by a hormone called erythropoietin. And erythropoietin is made by your kidneys, most of it, some by your liver. And that's the hormone that stimulates red cell production. So how it works is it gets the common stem cell that we at the start of the process, and it turns it into a red cell precursor. And once it's become that red cell precursor, it's committed into becoming a red blood cell, and it won't become a platelet or a white blood cell. It will be a red blood cell. And erythropoietin itself, its release is mediated by how much oxygen is in your tissues. So if you have low levels in your tissues, your kidneys are going to produce some more erythropoietin. That's going to hopefully produce more red blood cells and deliver more oxygen to your tissues. So it's kind of like a feedback loop. In terms of the structure of hemoglobin is what's in your red blood cells, and that's what's carrying the oxygen around to tissues. So your hemoglobin is made up of 4 chains. It's made up of 2 alpha chains and 2 beta chains, just chains with slight differences between them. What's very important about the structure is that it allows it to fulfill its function of transporting oxygen. So I put in a little box down the bottom that the bonds that happen between an alpha and a beta are a lot weaker than the bonds between chains of the same type. So that kind of makes a little door kind of thing so that your hemoglobin can change shape and make it easier for more oxygens to bond to it. So just in summary, a hemoglobin will have 4 heme groups or globin chains, and also each heme group has its own iron molecule. So in one hemoglobin, you have 4 heme groups and 4 iron molecules. So in terms of degradation, how it's processed through your body, your hemoglobin is broken down into heme and globin. So the globin part, because it's just a protein, it's going to be returned back to the body's pool of protein and amino acids. Your heme is the part of hemoglobin you're going to lose through either your urine or your feces. So how that happens is heme is going to break down into biliverdin, carbon monoxide, and iron. And the iron is going to be returned back to your bone marrow, and it's going to be recycled for new red blood cells. But the biliverdin, that is the part of heme that enters your urine and feces. You'll learn a bit more about that in year 2, and I don't think it's very important to know in detail. If you do want to know it, I guess you can just learn this diagram. But again, I don't think it's very relevant. And then in terms of binding affinity, so the hemoglobin and oxygen binding affinity is just how attracted hemoglobin and oxygen are to each other. Just one thing to note is that once one oxygen binds to hemoglobin, it becomes a lot more attractive to other hemoglobins. So pretty much it'll fill up with 4 oxygens after one has binded. So in terms of the factors that affect binding affinity, I've listed them there. I think the best way to think about it is the things that will happen when you exercise or increase metabolism. And that makes sense because if you have a high rate of metabolism and you're exercising, your body needs a lot of oxygen. So your oxygen needs to be able to leave your hemoglobin easily and enter your tissues. So things like increased temperature and then increased 2-3 DPG levels, that is just a byproduct of metabolism, and so is hydrogen ions and carbon dioxide. Those all indicate an increase in metabolism and will increase and will decrease how attracted the oxygen is to the hemoglobin, so that the oxygen can enter the tissues instead of staying attached. On the graph, you'll probably get asked a lot about right shift and left shift. So right shift pretty much means decreased affinity. So think about exercise and an increase in those factors that I've listed there. And then a left shift means a increased affinity. So they're more attracted to each other, and it'll just be the opposite of what I said. This is hemoglobin chain synthesis. I've put there that it's low yield because it's not very important at all. It's pretty much just saying that over development from conception to after birth, the types of chains that make up your hemoglobin will change. So in us and after birth, it's going to be 2 alpha and 2 beta. But as you can see on the graph, it's different before birth and it changes. And also the location of hemoglobin synthesis will change. So before birth, it can happen in your yolk sac, in your liver, in your bone marrow. And then after birth, anytime after birth, it happens always in your bone marrow. Just a little interesting fact is that HbF, which is the hemoglobin that's found in the fetus, has a greater affinity for oxygen than HbA, which is the hemoglobin in us. And that serves as a kind of like an advantage for the fetus so that they get oxygen. Okay, so iron enters the body through your diet, and it exits the body usually through the loss of skin cells, just natural shedding of skin cells over the day, and also menstruation, if you menstruate. And then it typically, the rest of it and majority of it will stay in circulation throughout your body in something called the plasma ion pool. And in that plasma ion pool, a lot of that will go back to your bone marrow and get recycled into new red blood cells. Some of that and any excess will go to your liver and get stored in your liver. Okay, so in terms of leukocytes and leukopoiesis, the process of leukopoiesis, or the formation of white blood cells, is that, so you have your hematopoietic stem cell at the very top. Those will split into your common myeloid progenitors and your common lymphoid progenitors. And as I mentioned before, your common myeloid progenitors are going to split into your mainly innate immune system cells. A lot of them are granulocytes. And then the other ones will split into, your common lymphoid ones will split into mainly adaptive immune system cells. The regulation, so what controls the process of hematopoietic cells becoming either one of myeloid or lymphoid progenitors and then its end cell type? That's controlled by 3 factors. So colony stimulating factors is the first one. They're just glycoproteins that stimulate leukopoiesis. That's pretty much the most in-depth you'll have to know for that. Also interleukins and immunological stimuli, which is allergies, bacterial infections, viral infections. With those last 2, it would kind of make sense that an increase in those in the body is going to promote the production of white blood cells, because interleukins will kind of indicate something like inflammation or infection happening. Immunological stimuli, like an infection, will also, I guess, indicate an infection. So in both of those situations, you would want your body to produce more white blood cells so you can fight it off. Thrombocytes and thrombopoiesis. So thrombocytes are also known as platelets, and what they are pretty much extensions of cytoplasm of these cells that are called mature megakaryocytes. Mature megakaryocytes are just large bone marrow cells. So you have the extensions of cytoplasm that break off and get released into the blood, and those are what are called platelets. Their key functions are in clotting. You will cover that in more depth in a few weeks, so don't worry about that now, but pretty much it's involved in adhesion, activation, and aggregation, which all help in the clotting process. In terms of how it's regulated, similar to how erythropoietin regulates erythropoiesis, thrombopoietin regulates thrombopoiesis. So that's a cytokine, is a cytokine as opposed to a hormone. And it's involved, as you can see in that diagram, along many steps of the way from the stem cell into a platelet. I've put a yellow box around them so you can see that pretty much every step is regulated by thrombopoietin, and especially the last step down the bottom where the platelets emerge from the mature megakaryocyte. So abnormalities that can happen with red blood cells, white blood cells, and platelets are here. I'll just go through some common like endings to words. So penia, if you ever see penia, that usually indicates a decrease in number. So we'll go through them, but you'll see like thrombocytopenia. So that's a decrease in the number of thrombocytes, or leukopenia, so a decrease in the number of leukocytes. And then you have cytosis and philia. Cytosis and philia usually indicate an increase in number. And the ending can alternate between those two. So if we look at erythrocyte abnormalities or red blood cell abnormalities, it can be categorized into pretty much 5 different categories. There's a decrease in number, which is usually termed anemia. Anemia is not just a decrease in red blood cells though, it also can be a decrease in hemoglobin levels. Then you can have an increase in the number, so that's erythrocytosis, cytosis being an increase in number. And that can be problematic if you have thicker blood that's less able to flow. It can often lead to clots forming more easily. Other abnormalities include the membrane. So with the membrane, there can be a defect in the structure of the membrane. You can also have autoimmune responses on your red blood cells if there are problems with the antibody reactions with your red blood cells. You can also have problems with the actual hemoglobin itself. So those chains that we talked about, there could be issues with them, or there could just be a decreased production, and that we saw was called anemia. Then there can also be issues with metabolism. So you can have deficiencies in the enzymes, and also issues with the metabolites and deficiencies in them. In terms of leukocyte abnormalities, it's very similar. So you have the decrease in number, leukopenia. So you can have a decrease in the number of neutrophils and in lymphocytes. And then you can also have an increase in the number. Usually if you have an increase in the number of lymphocytes, that's going to indicate an infection because your body is producing more white blood cells to fight off an infection of some sort. You can also have changes to the morphology. So you can have changes to nuclei and also the substances that are within the cell. Changes to cytoplasmic inclusions, which is the substances inside the cell, that can be quite problematic to the function of the cell. There are a few cells that are granulocytes, so they produce substances inside them and release them, and that's a part of their function. And so if there's changes to them, it can't really perform its function as it should be. So that's obviously a problem and can lead to certain diseases. Then thrombocyte abnormalities, again, you would have a decrease and an increase in number. And then you can also have changes in their ability to adhere, aggregate, and activate, so perform its function. So just lastly, there's some lab tests for the blood parameters. So there's FBEs and there's FBCs. An FBE is, an FBC is a full blood count. So that's what you see in the table there. And that table also shows the normal ranges of the different types of blood cells that you should be having. And an FBE is that table plus the histology picture that you're seeing on the right. The reason why that histology can be helpful in a lot of cases is that it can show you if there are any changes to the size, the shape, the appearance, or any intracellular components of the blood cells. So it can just provide some additional information. And obviously, if you notice any changes from the normal range, it can indicate some sort of pathology or infection.
Sounds good. We'll see you guys next week. Thank you.